Hello, my name is Dr. Liron Bar-Peled, and I'm an assistant professor at Harvard Medical School and a member of the Massachusetts General Hospital Cancer Center. In our lab, we seek to challenge the conventional notions of what is druggable in cancer. This is critical for cancers such as cholangiocarcinoma, because while advances in cancer genetics have identified potentially actionable genomic alterations in roughly 45% of patients with this disease, for the remaining 55% of cholangiocarcinoma patients, there are no rational next-line therapies after tumors show progression following chemotherapy. Now, it's important to understand that most cancer drug development has focused on targeting proteins with enzymatic activities, such as kinases, whose chemical structure is considered readily susceptible to drug inhibition. And these types of proteins are often referred to as druggable. However, after many years of cancer biology research, we've identified a great deal of additional genes that are essential for cancer growth, but are not currently being studied as viable candidate drug targets because they are not conventionally considered druggable. We believe that an effective assault on cancer requires the implementation of creative strategies that allow us to expand our arsenal of drugs to include these important but conventionally undruggable cancer genes. To this end, we have developed a suite of cutting-edge technologies in our lab that combine advances in chemistry and proteomics and allow us to rapidly identify druggable proteins in cancer cells. Specifically, we are targeting one important component of a protein, the amino acid residue cystine. Cystines play critical roles in protein function, and because of their importance and unique reactivity, they are the target of multiple clinically approved drugs. We recently developed cystine drugability mapping, which defines the landscape of protein drugability in native biological systems. Now, classic drug discovery campaigns focus on one protein at a time and require costly and time-consuming high-throughput chemical screens. CDM allows us to bypass these conventional approaches by immediately nominating proteins with druggable cystines. This enables us to screen targeted small molecule libraries and rapidly identify first-in-class chemical inhibitors. Using CDM, or cystine drugability mapping, we are essentially making a drugability map, or drug map, of cancer cells. Now, this is incredibly important because this drug map serves as our guide or atlas as we develop small molecule inhibitors to target key cancer drivers. With the help of the Evan Schumacher Fund for Cancer Research, we are testing the hypothesis that protein drugability in cholangiocarcinoma has been vastly underestimated, and that many classes of proteins, including cancer drivers, can be targeted with small molecule inhibitors. To test this hypothesis, we are making drug maps of cholangiocarcinoma. In collaboration with my colleague and good friend, Dr. Nabil Bardisi, we are first using patient-derived cell lines to map druggable proteins in cholangiocarcinoma. We hope that soon we will also be able to make these drug maps in tumors directly taken from patients, so that we will be able to make a personalized drug map for each tumor. In the second phase of this project, following the establishment of these cholangiocarcinoma drug maps, we will then integrate advances in functional genomics to identify druggable cancer drivers. We will use this information to then screen our advanced drug libraries in our lab and develop hit compounds into potent leads. So without the Evan Schumacher Fund, we simply would not be able to undertake this exciting research on developing new drugs for cholangiocarcinoma. With this flexible seed funding, we are able to undertake targeted experiments that would not be supported by traditional grant mechanisms. And I truly believe that if successful, this project could be a launching pad for the development of a new class of inhibitors specific to cholangiocarcinoma. Now, I want to say that I'm incredibly humbled to receive this grant, and I hope that in some small part, our research may honor the memory of Evan Schumacher. Thank you once again for your incredibly generous donation, and I'd be happy to follow up on any of these points.